Hello everyone, this is Medical Gateway, and now we are studying the last part of chapter number 3. In this part, we'll study mitosis, and other than the mitosis, we'll also discuss very important topics like telomeres, apoptosis, necrosis, and cancer. So let's start with mitosis. Okay, so what is the life cycle of the cell? So the period from one cell reproduction to the next cell reproduction is known as the complete life cycle of a cell. So what is mitosis? Mitosis is the series of events that cause division of cell. So mitosis is basically the cell division. So, but if someone asks you to define it, then it is basically the series of events that cause division of cell. Okay, now what is interphase? Now, the interval between two mitoses is known as interphase. So, the interval between two mitoses is known as interphase. So if you look here, then it means mitosis plus the interphase makes the complete life cycle of the cell. Okay, so mitosis along with the interphase makes the complete life cycle of the cell. So 95% of the life cycle of the cell is basically interphase, the phase between the two mitoses, whereas mitosis is only 5% of the life cycle of the cell. And what is the duration of mitosis? Mitosis takes place in 30 minutes when it is started. Okay, now when exactly the DNA is duplicated before mitosis? So DNA is duplicated 4 to 8 hours before mitosis. Okay, and when the duplication of the DNA is completed, after that, 1 to 2 hours after that, the mitosis begins. So the cell is in interphase, then interphase the period between the two mitoses. Then when the mitosis is going to start, before that, 4 to 8 hours before that, the DNA will be duplicated. And when the DNA is duplicated, 1 to 2 hours after that, mitosis will take place. Okay, so in mitosis, basically how the DNA is duplicated. Now this is the DNA. The two strands of the DNA will split into single strands, and both the strands will act as a template for another strand. Okay, so in this way, one double-stranded DNA is converted into two double-stranded DNA. Okay, now when the mitosis will take place, each of these double-stranded DNAs will go to a separate cell. Okay, now let's discuss a few other important things regarding the mitosis. Now, what is the significance of DNA polymerase and DNA ligase in the mitosis? Okay, as we all know, DNA polymerase is a very important enzyme which will cause the mitosis. Okay, now what is the function of DNA ligase? Now, DNA ligase will create bonds between successive nucleotides, and it will also make bonds between the two strands. Now, this whole is a single strand. Now, when the new nucleotides are attached along with it, the respective nucleotides have to form bond between their neighboring nucleotides. So that bond is formed by DNA ligase. And when the strand is completed, this is a single strand, another strand is completed here. Now these two strands have to form hydrogen bonds between them. These hydrogen bonds are also formed by DNA ligase. Now, when this duplication of the DNA is completed, after that, there is proofreading of this DNA to check whether the DNA is replicated in the correct manner or not. So that DNA proofreading is also done by them. And finally, if there is a problem in the DNA duplication, then DNA repair is also done. Okay, So this is what we have discussed, the significance of DNA polymerase and DNA ligase. Okay, then what are the histones? If you focus on the diagram here, there are blue dots in between the two strands of the DNA. Specifically, these blue substances are located where the two strands of the DNA are overlapping each other. So these blue dots are known as histones. So histones are basically round proteins. The main function of histones is that the DNA helix coils around the histone. Okay, so this is one strand. When the other strand is formed, they both are coiled around each other. So this coiling of the DNA takes place around the histones. Okay, so this is the significance of histones. So now let's discuss the stages and the phases of mitosis. So first phase is the prophase. So in prophase, there are two things that are happening. One is that the chromosome will be converted into well-defined form. Okay, so initially the chromosome, this green one inside the nucleus, is not in the well-defined form. But in prophase, when the mitosis begins, this chromosome will be converted into this well-defined typical form of the chromosome. The second change in the prophase is, which is the first phase of the mitosis, is that there will be spindle formation in the centrioles. Okay, so these are the two centrioles, and the centrioles will show spindle formation. Okay, now what is the significance of spindle formation? We'll discuss it in the next phases. Then we have prometaphase. Now in prometaphase, one thing is that nuclear envelope will disappear. If you focus here, there is a nuclear envelope in prophase, whereas here in the prometaphase, the nuclear envelope has disappeared. And the microtubules elongate, and they are attached to the centrosome, the central part of the chromosome. So one in prometaphase, first thing is that nuclear envelope disappears, and the second thing is that microtubules get attached to the centrosomes present in the center of the chromosomes. Okay, then we have, after prometaphase, we have metaphase. In metaphase, what is happening, that these microtubules pull the chromosomes, the chromatids, in the center of the cell, and they are arranged, the chromatids are arranged in the center of the cell, and they form an equatorial plate with the help of mitotic spindles. Okay, so in metaphase, these spindles will pull these chromatids to the center of the cell and arrange them in a line, forming the equatorial plate of mitotic spindles. Then we have anaphase. So in anaphase, what is going to happen is that these microtubules will pull these the pairs of chromatids apart. Half of the chromatids, the chromosomes, will move to one side, and half will start moving to the other side. So this is going to happen in the anaphase. Then we have the telophase. So in telophase, what is happening is that now the job of the mitotic apparatus is completed. So the mitotic apparatus starts to disappear in telophase, and the nuclear membrane starts to appear. Now these chromatids, which are being pulled to the two sides, now nuclear envelope starts appearing around these chromatids like this, and finally the nuclear envelope will be completed in this stage. And finally, the most important thing, cell pinches in two cells. Now there are contractile proteins in the cell. These contractile proteins will contract and pinch the cell from the center, dividing the cell into two. Okay, so by this, the mitosis will be completed. The cell will split into two cells, and both the cells will have the complete genetic material, as initially the chromosomes were divided into two. Okay, so the phases are prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, and telophase. And the phase between prophase and metaphase is prometaphase. So pro from prophase and meta from metaphase. So prometaphase. Okay. So in the first phase, basically, the chromosome is well defined and the spindle starts appearing. In the second phase, the nuclear envelope disappears and the microtubules are attached to the centrosome. In the third phase, the chromatids are pulled to the center, forming an equatorial line. In the fourth anaphase, the microtubules pull the chromatid towards the two sides. And in the telophase, the mitotic apparatus disappears, the membrane starts appearing around the chromatids, and the cell pinches into two with the help of contractile proteins. So this is how a single cell is divided into two cells. So this is mitosis. Now let's discuss telomeres, a very important topic. Telomeres, and a very interesting one. Now, what exactly are telomeres? So this is a chromosome. Now, if you focus at the end of the chromosome, there are telomeres. Now, what exactly are telomeres? So if I zoom on it, so these are the telomeres. These are the telomeres. The gray colored ones are the telomeres. So what exactly are the telomeres? These are repetitive nucleotide sequence at each end of chromosome. So at each end of chromosome, there are a repetitive sequence of nucleotides, which are known as telomere. So this is the telomere, and this is the telomere, and this is a telomere, and this is also a telomere. Now, telomeres act as protective caps for the DNA. Okay, so if this telomere is decreased in size, for instance, one DNA is this and the other DNA is this, if the telomere size is decreased, it means now the protective function of telomere for the DNA is reduced, and this DNA is now prone to damage. Okay, and the interesting thing is that with each DNA replication, the size of the telomeres is reduced. So with each DNA replication or with aging, the size of the telomere is reduced and the DNA becomes more prone to damage. So this is what aging is. So telomeres have a huge amount of significance with reference to the aging. So telomeres means aging. So decrease in size of telomeres is basically aging, because telomeres are protecting the DNA. Now, how exactly with aging or with each replication of the DNA, the size of the telomeres is decreasing? Now, with each DNA replication, basically few nucleotides from each telomere are lost. So with each DNA replication, few nucleotides from each telomeres are lost. And why this is happening? Because of missing of few nucleotides by RNA primer attachment. Now, as you know, whenever the replication is taking place or the RNA is being formed, so RNA primer is attached. So when the RNA primer is attached, it will miss few nucleotides every time a new DNA is being formed or an RNA is formed from the DNA. So this will result into missing of few nucleotides every time the replication is taking place. So in every division, the telomere size is decreasing, and as you know, the cells are repetitively dividing for the whole life. So with aging, the size of the telomere is reduced to a smaller size. So the telomeres are now unable to protect the DNA from damage. So that's why this is the significance of telomeres in aging. Okay, so this is the function of telomeres. Now let's discuss the function of what is necrosis. Now necrosis is basically whenever a tissue suffers from injury, so the tissue swells and the tissue bursts. When the tissue bursts, there is spillage of the content of the tissue or the cell out of the cell, and when the content of there is spillage of the inside damaged dead content of the cell, it results into inflammation. So necrosis is basically cell death due to injury, and this cell death is not a programmed cell death. This cell death is happening because of injury. Okay, so whenever the cell suffers from injury, it swells up, it bursts, it spills its contents outwards and resulting into inflammation. Now I have mentioned here it is not a programmed cell death. Now, is there a programmed cell death, and why the cell is going through a programmed cell death? Yes. Apoptosis is programmed cell death. This cell death is not because of injury. This cell death is meant to occur. For instance, a new cell is formed by cell division, and that cell is itself programmed to die after a few months or after a few years. So this is known as programmed cell death, and then that cell will die and it will be replaced by a new cell. So this is known as apoptosis, the programmed cell death, whereas necrosis is not a programmed cell death. This is cell death due to injury. So what happens in apoptosis, or how a cell programs its own death? For instance, if the cell has to live for a few months, then after a few months, procaspases inside the cell will be activated into caspases, and these caspases will cause cell to shrink, condense, and the inside cytoskeleton of the cell will disassemble. Okay, and when the inside of the cell shrinks and becomes condensed and the cytoskeleton is disassembled, then the cell surface will also be altered. There will not be any spillage or the inflammation in this case, just the cell surface will be altered. Okay, and when the cell surface is altered, the cell surface is not normal, the cell surface is not smooth, then the neighboring phagocytes will recognize this cell as some dying cell, and the neighboring phagocytes will eat this cell. Okay, but there will be no spillage, no swelling, no inflammation in the case of apoptosis. Procaspases is converted into caspases, which will shrink the cell surface will be altered, the neighboring cells recognize them as the dying cells, and the neighboring phagocytes will come and eat that cell. This is how apoptosis is done. Okay, so, and in the last we're going to discuss cancer. Now, what exactly is cancer, and what are the cancerous cells? So cancer is basically a condition in which there is uncontrolled multiplication of some abnormal cells in the body. For instance, if one cell has to divide into or multiply into 5 cells, in cancer that cell might multiply into 500 or 5,000 cells. So this the condition in which there is uncontrolled multiplication of cells, some abnormal cells, this is known as cancer. Now, what exactly causes cancer? So cancer is basically caused by some hereditary tendencies, and then there are some external factors which are predisposing factors for cancer, which include the chemical substances, some cancer-causing chemical substances, some physical irritants also cause cancer, and then ionizing radiations also cause cancer, and then some viruses also can turn a normal cell into a cancerous cell. So hereditary tendencies, chemical substances, physical irritants, ionizing radiations, and viruses, these are the causes for cancer. Now, there are 2 questions in the Guyton. Why only few mutations lead to cancer? There are a number of mutations taking place inside the body, but still, as compared to the number of the mutations, the number of the mutations converting into the cancerous cells are very low. Why is this the case? So the answer is, any mutated cell or most of the mutated cells have less survival capability. And why is it so? Number 1 is the mutated cells. For instance, if a cell which was initially normally programmed to multiply into 5 cells is multiplied into 5,000 cells. Now all these cells will require extra nutrition, but there is no extra nutrition for these cells. The nutrition was supplied for just 5 cells, but there are now 500 cells. Now these cells will die because of the inavailability of the nutrition. Secondly, the immune system of the body blocks these cells, or immune system of the body recognizes these cells and destroys them. And then there is a normal feedback control of the cell reproduction, which also takes place. So these are the factors which will lead to very less number of mutated cells to be converted into the cancerous cells. Okay, now, but still, some mutated cells will be converted into the cancer cells. And why is it so? And what are their characteristics? So first thing, I have discussed here that they need excessive nutrition, which is not usually supplied. But in some of the mutations, some of the cancerous cells are able to survive because they themselves secrete angiogenic factors. They themselves secrete angiogenic factors, and these angiogenic factors will result into numerous new blood vessels developing for them, supplying them extra amount of energy. So this is one thing that why the cancer invasive which is included in the invasive characteristics of the cell. Number 1 is angiogenic factors. They release angiogenic factors, developing new blood vessels, and those new blood vessels are supplying extra amount of energy to these invasive cancerous cells. And the second point why they are the invasive why some of the cancers are invasive is that these cancer cells are less adhesive. Normally the cells are adhesive, they stay at their position, but cancer cells are less adhesive. They're less adhesive, they might go into their neighboring tissues, they might go into the blood vessels and travel from one point to another, and by this they metastasize from one point to the other. Okay, and thirdly, the invasive cancer cells does not respect growth limit. For instance, all the normal cells are programmed to grow in a limited fashion. They just cannot grow or increase their size to an uncontrolled level. But cancer cells does not respect the growth limit. So there are 3 points why regarding the invasive cancer characteristics. One is they do not respect growth limit. Second one is they bring their own angiogenic factors with them. They secrete the angiogenic factors, developing the blood vessels. And thirdly, they are less adhesive, by which they can easily metastasize from one point to the other. So by this, we have finished third chapter of the Guyton. See you in next chapter.